So my talk, would be on prostate cancer systems genomic analysis, which we have been doing for over the last 2.5 years. This was a kind of a grant, that a seed grant, that we obtained from BrainPan, for which I'm as well an advisor. So my talk, would delve on the rationale behind this prostate cancer project. Where are we now, and what are the kind of problems and challenges, and the kind of pilot study that we have done, taking into the whole exome sequencing capture with respect to the samples that we have taken, and the kind of mutations that we have found in high-grade tumors and their inherent pathways. And since the conference theme is on data analytics or data heuristics, a small component, I would probably delve on the data science heuristics that this project would cater upon. And finally, the kind of future projects, especially taking the long non-coding RNAs and the regulatory elements that we would work on. So I come from Birla Institute of Scientific Research. This is a not-for-profit research institute. Our academic setting is based in Birla Institute of Technology, Mestra, which also has got a kind of a campus in Jaipur as well. And our current chairman is C.K. Birla, and our current executive director is Dr. Punendra Ghosh. And ours is headed by Dr. Krishna Mohan, who is the head R&D. And we are a group of about 14 scientists, about 7 PhD fellows and 2 postdocs now, working at Birla Institute of Scientific Research with 2 major laboratories. One is biotechnology and bioinformatics, and the other one is remote sensing, where we take geospatial satellite imaging to collect the flora and fauna, and do some kind of an exciting research. So my talk would delve into 3 small stories. So the first part is about the prostate cancer and the kind of mutations relevant to India. It is unfortunate that prostate cancer is burgeoning in India. It is the 3rd most prevalent cancer in India, and it is absolutely burgeoning with as many as the Globocon statistics saying that the current Globocon statistics and the population-based cancer registries, giving a kind of an alarming incidence rate. So overall, if you really see, 1 in 9 men are usually diagnosed with prostate cancer during his lifetime. So it's a male organ-specific disease. So it's usually caused with people aged above 55 years. And it also leads to the so-called declining fertility rates, which is counter-effective with respect to the prostate cancer tumors that are uprising right from benign cases till the malignant cases. If you really see the rationale behind this particular project, in India there are so many prostate cancer cases, but we don't know what are the kind of inherent mutations that are relative to Indian subpopulation. And furthermore, India is considered the diabetic capital, and we were very much interested if diabetes is really relative to prostate cancer, or the other way around, whether or not prostate cancer victims would have an inherent chance or likely chance to be diabetic in their lifetime. So while I was doing my 4th postdoc in 2014, one of my colleagues, he was working on mice and as well as cattle and pigs. He would overfeed the pigs and cows so that they would become obese, and we would probably isolate these animal tissues, the fat tissues, and check for the relative expression of the genes. So he would overfeed the cows, he would overfeed the pigs, he would overfeed the mice. And then what happens? That was a kind of troubleshooting. The lab was working on in Hajar Kadamudeen's lab. And as a result, many of these overfeeding trait efficiency would also result not only in diabetes, the type 2 diabetes, but also some of these particular cases were also prone to prostate as well. Because the animals are poor, we don't really do a lot of research in identifying whether or not an animal is a prostate victim or not. We're not really bothered. But if you really take this analogy in humans, it's absolutely the same case. Because the research that was carried out on diabetic pigs, which were prostate victims, could be extrapolated to the humans as well, because the human rumen is also something similar to the pig rumen. Okay, so the entire digestive system, the entire tract, right from the neck till the legs, it's almost the same. Only thing is, pig walks on 4 legs, and the humans, walk with 2 legs. Okay, so having said this, that is the rationale behind that. And if you see the prostate cancer cases in the far west or in the Caucasian population, that is because of due to meat eating people. Many non-vegetarians are prone to prostate cancer. That is a well-known fact. So which means that there is a diet-induced factors, a diet-induced obesity, a diet-induced constraints because they eat a kind of half-cooked red meat. But in India, at least from the kind of the samples or the cohort that we have identified, 98% of the samples are all vegetarians, and yet we have got prostate cancer. So that is really alarming. So that begs us a question: why is that Asian Indians who eat a lot of vegetarian stuff are really related to, or really getting this prostate cancer tumors? So it is also associated with vitamin D factors as well. Although we get a lot of sunshine, we drink so-called milk, but the milk also harbors some kind of vitamin D, vitamin D in it, and yet there are some cases prostate victims. There are also some benign cases. Some of the renal or urorectal cases that are absolutely benign, that are not at all prostatic, like fistula, piles, none of them are really getting the prostate cancer at all. There are hardly very few cases, there's no correlation at all. But the people who have not been diagnosed with these particular cases in their lifetime, or who might really be diabetic, might be associated with prostate cancer. So that begs us a question that if you really see in terms of the economies as well, there are countries like US, UK, Western Europe, the whole of Western Europe, a lot of prostate cancer cases are seen because they're all high per capita income countries. And usually from the diagnostic front, from the clinical front, many entities are taken. There usually something called the distal rectal examination is taken. There is something called the prostate serum antigen. And of course, we also take the biopsies. There is also the TNM grading, where we see the number of lymph nodes associated with the tumor, and whether or not those lymph nodes are really leading to any metastatic condition that can also be inferred. So in the earlier cases, in the earlier urogenital cancers, there were some well-known markers like the HK3, which is a kallikrein gene markers, which have been absolutely deciphered. But unfortunately, these particular markers are not really taken in analogy with prostate as such. For some reasons or the other, it has not been that sensitive. MALAT1 is one of the favorite long-known coding RNAs that has been considered as a urinary biomarker. So what we did was, our total cohort is about 120, getting this prostate cancer FFP blocks and radioprostatectomy samples is certainly a huge task. So as a pilot, we started with 10 samples, another 43 are on anvil. We might get results pretty soon. And we have taken the ethical clearance, and these are all the cases. We have taken the 5 benign prostatic hyperplasia, and 2 with intermediate Gleason scores of 6 and 7, and of course 3 with Gleason score above 7, which are so-called the high-grade tumors. In fact, Gleason score above 8 from the diagnostic or from the clinical front is called as the high-grade score. And this is the inclusion and exclusion criteria. So for the malignant cases, we have taken Gleason score above 6 with PSA 4 nanogram per ml or above. And for normal or benign prostatic hyperplasia, which is Gleason score 6, less than 6. And some of the samples are also extrapolated for RNA-seq, which we're also trying to work on. And we set up our own pipeline, which is our benchmark pipeline, which we established in 2017 and published in 2018, as you could see, which is Meena et al. And we used all the well-known whole exome sequencing and ACMG guidelines to infer and call the variants. And we also got good read size, which is about 9 GB with an approximate depth of coverage of above 90x coverage for all those particular samples. And more samples are also on anvil. So on an average, we got about 9.8, which is absolutely expected. So this variable grading, as you could see, this variable grading could also be extrapolated to vegetarians. As you could see, almost all of them are all vegetarians. Maybe one, when we counsel them, he said that he was a non-vegetarian at some point of time, but he's no longer a non-vegetarian. So that was some years ago. So now coming to the results, we know that in case of prostate cancer, you get a lot of genes. There are 30 well-known genes, in fact 33 well-known genes, and of course we found mutations across all those the so-called 30 well-known genes. As you could see, the square boxes are all those particular mutations seen across those particular genes from the Caucasian population, whereas the ellipse or elliptical ones are all from the Indian population. As you could see, these mutations, the CTNS mutations, are absolutely pathogenic, and they are inferred to be associated with vitamin D as well. Whereas there are some mitochondrial and as well as obesity associated genic mutations coming from OPA1, which are also influencing our Indian population, which begs us a question that there could be patients who are obese and who are prostatic, and they might have this asphyxiation problem because of loss of this mitochondrial deoxphos pathways in them. So it's just like this. The theory here is if the person is obese and if the person is prostatic, then you will have fewer mitochondria because they cannot really breathe properly, and that really leads to the so-called the loss of tumor cells, or the loss of healthy cells. I'm sorry, not the tumor cells. So we did this, and we have taken the depth of coverage with above 5 and as well as 20. These are the wonderful plots now made by Ayam and Nidhi, my PhD fellows over here. And we found some inherent mutations specific to Indian conditions. And this is what we published last week. And this was a kind of a recent paper, thanks to wonderful collaborations and samples, that were collated from my clinical collaborators, Devendra Sharma and Manish Vijay, who are reputed urologists working for Rukmini Birla hospitals. So the first part of the talk here is we knew that most of the patients in our cohort are vegetarians and yet prostate victims, and we began to understand whether or not any obesity associated risk, polygenic risk is associated with them. And not only obesity, but also diet or mitochondrial dysfunction and also vitamin D deficiency. And we also found some rare disease events associated as well, like Usher syndrome, which is more related to immune prophylaxis. And these immune prophylaxis is related to the regulatory events associated with those particular patients. And of course, the well-known other tumor suppressor, which is P53 as well. So these two are well-known tumor suppressors. And incidentally, COL6A1 has got about 7 copy number variants, and we found one important copy number variant that is seen in the high-grade tumor, which we could really detect, from our pipeline. And the diet-induced obesity, the OPA1, which is a well-known candidate, it's really seen, and we got 3 pathogenic and 4 likely pathogenic mutations seen across our Indian cohort. And of course, the CTNS. So now we have some data, to move forward, where we have developed a kind of a conceptualized framework of mutations, where this particular data can be further used for prioritization. So now we begin to ask, now that we know what are the genes responsible for prostate cancer in India, now we also know the mutations. Many of them are pathogenic and likely pathogenic and bona fide. And now we have our own story to work on, which we call it as a genome walking story. So it is like this. So we know the genes responsible for prostate cancer, and we know where mutations are. So for example, this is a BRCA2 gene context, the highlighted one, which is in pink color, as you could see. And our mutations are somewhere over here, all through the length and breadth of this. And what we usually do is, these are all the intergenic regions. As you could see, these are all the intergenic regions. So we might even have the non-coding RNAs coming between them. But what we preferentially do was, since this is a BRCA2 gene, we further walk 5' UTR, the untranslated region, where we have a lot of transcription factor binding sites, the CTCF motifs, the long non-coding RNAs, the enhancers, and most importantly, the so-called open chromatin elements, which fall up to 5 kb, but in a rare case, you also find, some of them, falling, up to the 10 kb region, masking the gene of your interest. So we asked how many of these particular regulatory elements impromptu are known to be associated with the genes causing with prostate cancer in India. Of course, BRCA2 is a well-known figure. It's also seen in Caucasian population as well. So as you could see here, so all these are all the long non-coding RNAs in boxes. The circle ones are the CTCF binding sites. And of course, you've got the enhancers, which are in yellow color as well. And this is a rarity. You also find the enhancers associated with that. So we have annotated them to the fullest. We know what are those long non-coding RNAs and long intergenic non-coding RNAs. We use the Smith-Waterman algorithm. We develop a kind of a bidirectional best blast hits. We have all those particular things in our pipeline. And these are all the wonderful figures developed by Nidhi and Narmada. Narmada was a graduate student. And we isolated those long non-coding RNAs. And now our experiments are on the anvil, where about 10 long non-coding RNAs have been identified with a respectable absorbance of 1.9, 860 by 280. And now we are trying to ask whether or not these long non-coding RNAs are known to be interacting with the proteins in question impromptu. So this is how. So this, as of now, we have taken these two. One is a long non-coding RNA non-exact 053810, which is associated with BRCA1. In fact, there's also BRCA2 gene as well. BRCA2 non-coding gene, which is non-exact 103724.2, which is somewhere over here. And we ask whether or not these are all the sure shots, because many a time there's a chance that if there is a non-coding RNA sitting upstream of that particular gene, many a time it has got highest per chance to interact. And we use some tools like LinkPro and RPFRED and reach consensus whether or not they are interacting. And our experiments, the physical experiments in the lab, we are just trying to check their expression and as well as perform some kind of pull down assays. Interestingly, in cancers at least in prostate, we don't no one has really identified the pvRNAs. In fact, pvRNA, one candidate also we found this to be associated with COL6A1, which is a kind of a copy number variant, which I was telling you last time. So on a different note, COVID has given us a kind of a blessing in disguise. So we have worked with Keshav Singh's laboratory, where we identified that in COVID samples, we also find some of the non-coding RNAs that are associated in COVID positive patients. That indicates that since prostate is a male tissue, there is a likely chance that the males are more prone to be associated with COVID samples or COVID infection. And they're also more likely to be, there's a chance that the prostate victims are more likely to survive with COVID positive infection. So in that particular process, in that story, we also have these non-coding RNAs, which we also considered them to be the candidates. So the part 2 of the conclusions, we have the pvRNAs also identified. The pvRNAs are small non-coding RNAs. Unlike microRNAs, they're bigger than microRNAs with almost one-fold. They're anywhere between 21 to 35 nucleotides, unlike the microRNAs, which are maximum 24 nucleotides. And pvRNAs are also highly conserved, but not that conserved when we consider in relation to the microRNAs. And they're also known to be interacting with the targeted RNA and aid in regulation. And the long non-coding RNA protein interactions and the pvRNA protein interactions, which I described using our annotation, is on the anvil. And there are also some sets of pvRNAs, some bases of pvRNAs, which are impromptu interacting with the proteins of your interest. They could be any proteins. In this case, the BRCA2 example, which I was taking. So they play a wonderful role. So those RNA are called as the Fuzzy RNAs. So Fuzzy RNAs are subclades of the pvRNAs, and they also play a very important role for aiding regulation. And they could be the wonderful plausible markers on anvil. So the final part of my story, so now we have huge data, we have got huge annotation, we have got about 120 data sets, 120 samples, coming sometime in a couple of months, and we have prioritized the SNPs. So on an average, if we assume that there are 120 samples, taking up to 200 samples or 200 SNPs, including the non-coding RNA SNPs as well, because I only described about the whole exome sequencing as well. But the part 2 of the story is we also have the RNA-seq, which I didn't really describe much. So we have about 24,000 variants. And of course, we need to validate them in the lab. So let's assume 5% as per the statistical significance, including the housekeeping genes. So that begs us to a question that we need to validate about 1,200 samples. And among these 1,200 non-coding RNA SNPs, if we really need to go for Sanger validation, it would be pretty expensive. So this is where our part 3 comes into picture. We really don't want to sequence, prostate cancer samples from 121st exome or 121st transcriptome. We are done. We are good for done now. We only want to ask, we have huge data sets right now. Can we take this into a better machine learning training approach? Can we really train them on a network of prioritized SNPs and ask, hey, look, we have so many samples in one column, and across the samples, we have got the SNPs pertaining to, diabetes, the vitamin D deficiency, blah, 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 blah. And now, can we use something called deep variant to extrapolate our approach, our predictions, and ask whether or not, if at all, if 121st sample has got any of these particular disease phenotypes, can we say that in those particular patients we have, this mutation, that might be predicted. Okay, so the end goal is here to develop a kind of a panel. That's where, the prostate cancer panel would really be useful. There's no panel developed for prostate cancer, taking in all these long non-coding RNAs or pvRNAs, as such. So our study is really approaching towards that, taking these particular, wonderful pictures, looking into the regulatory aspects. So this is a problem which I was really describing. Just for a short of time, I'm just moving ahead. That would really give us a kind of a huge incentive, because if you really look into the prostate cancer cases in the West, people spend huge monies, even although in India, the clinical market is pretty high, even, for a simplest biopsy, as you could see here, the cost is pretty high. In US, it costs about $2,615. And even in India, for a simplest biopsy, it costs anywhere between 60,000 to 80,000 rupees, which is quite high. Do you think that we Indians with a stigma, where we have some pain in our genitalia or in scrotum, we would approach a doctor? I doubt, we'll not be able to approach a doctor. I mean, maybe even me as a scientist, probably I would probably at least think twice. So that's where, we wanted to develop a kind of a simplest pregnancy-like kit, taking a simplest urinary-based biomarker, taking this long non-coding RNA as a case with all these particular conditions, taking into picture. So the deep variant results I've analyzed for about 40 samples, taking the big data sets as well. But my group members, Ayam and Nidhi, they are currently analyzing, taking the current scenario into picture as we get more and more sequencing data. So the final lessons. So we have prioritized the long non-coding RNAs and one pvRNA. And the case here is we wanted to identify those regulatory bells or regulatory elements that are present in the 5' untranslated region, and see if any of those candidates would have an influence in the way the mutations are seen in these particular patients. So which means, there could be candidate interacting partners associated with this regulation, thus aiding a complete change in the way the mutation is heading. So the present results, with respect to multi-gene panel testing, we could probably add our coding and non-coding SNPs across the various disease phenotypes. And our immediate goal as of now is also take the Gleason score attributes across all those 120 samples. And the distant goal is to look into the hereditary prostate cancer. I doubt, in India, if we can really do the hereditary prostate cancer things, or the germline prostate cancer in the picture in the near future, because we started just getting, these particular samples, taking out of this stigma. And of course, the whole exome sequence data and phenotypic variables. As of now, the sad story here is there are about 18,000 prostate cancer exomes done all over the world. And 99% of the samples are not uploaded in GenBank. They're all part of consortium. They're all folks are working with various companies for developing certain panels or developing, some drugs. That's a very unfortunate thing. If we have at least 10% of those particular samples, 18,000, 10% would come to about 1,800. We would have really been a myriad out of it, where we could have taken our machine learning approach to a better scalability. So the role of factors like diabetes, obesity, vitamin D, and dietary induced nutrient intake is absolutely associated with prostate cancer. There could be genetic factors, which we are trying to check with the IndiGene network that Vinod Skari has established. There are also the changes that we could also see across the long non-coding RNAs as well, the SNPs, and as well as the structural rearrangements, including the copy number variants. And coming up, we are also working on the RNA-seq, as I told you, and the long non-coding RNA screening and long non-coding RNA protein interaction studies for selective biomarker studies. And of course, we wanted to come up with something like MALAT1, taking all these conditions, including the hemorrhoids as well in the near future, where we are working with collaborators. So that's where our systems genomic picture come into picture, coming to case, coming to a valid example. So we don't just do research. We have got a wonderful group here. We have Nidhi Shukla, who is a women scientist, and Ann Gupta, who is a PhD fellow. So both of them are doing PhDs on prostate cancer with me. And of course, Ayam is doing with prostate cancer and vitamin K deficiency. We also counsel the patients. So this picture is taken after due permission from the patient himself. So we also counsel, once in 6 months, talk to them and also teach them, the science behind the tumors. So finally, I would like to thank Abhi and Pankaj and the entire the conference team for your kind invitation. I mean, no words of appreciation would be enough, for Nidhi Shukla and I am Gupta, who are wonderful PhD fellows in making. My lab, my group, doesn't go without these, two appreciations. And three more PhD fellows have joined with me: Shalini, Buman Kaur, and Anchita. Arshita is my PhD fellow who is working on recommender systems where she will work on these prostate cancer things. Purva and Sonal are guest postdocs, and Narmada was the one who was doing the graduate fellowship with me. She did an absolute work, staying for 3 months here. And Mamta Nehra was the other one. Thanks to wonderful funding from BrainPan, Amrit and Saurabh, and Devendra Sharma and Manish Vijay from RBH, KM, my mentor, head here. Obal is my collaborator. Praveen Mathur is another pediatric urologist. Ashwini, my good friend, who is from DNA Experts, where we do a lot of sequencing, and other people over here who are really making, making me happy every time, with a lot of discussions. And please join BioClues. I'm the founder of BioClues, and Abhi is also a member of it. Is a core member. Is a managing editor for IJCB, the journal that we are associated with. So I look forward for some kind of questions. Yeah. Thank you so much.